on algae and fish waste. So, as I told just before, I go around all the advanced science as well as the basic and applied sciences. Due to some reason, departmental reason, and as well as the student lacuna, as well as the funding lacuna, as well as the facilities in Yoga University, I planted best research in economic way of funding. Say for example, with low amount I need to go with it. You must say thanks to DBT that government is always spending money for us to motivate ourselves to go deep in research. Wherever you are there, either it may be biotechnology, zoology, or botany, or environmental science and material science, and wherever you go there, but you need to focus on deep, not the deep. Like say for example, go deep and search what is real output of this research and what best we can do for this society. That is the only way. Everybody say, and people are claiming one science to other science, even within botany or within zoology, they say that this research is good, this research is bad, and all. That is not criteria now. Government is seriously looking for what is your output, what is going to be, you are going to give, what product you are going to give to the society. That is the only concept now. And still we are fortunate enough that we are still getting funding for this kind of programs. So anyway, I'm going to start this introductory or funding business. I'm going to talk about my topic and I told you that why I came here. So biohydrogen and bioethanol and biodiesel production from algae and fish waste. So how many people know that the algae? What is algae? Telugulo chakkaga. What is algae? బాగా రా తెలుగు నాకు ఈ సాయిబలాలు దానికంటే రా తెలుగు పాచి అవునా వై వీఆర్ బై సీయింగ్ ది పాచిని చూస్తే ఎందుకు హెసిటేట్ చేస్తాం ఆ పాచి నీళ్ళలో కెలిగిన చేపను తింటాం మళ్ళా అవునా వై సో ద థింగ్ ఈస్ హియర్ ద సైన్స్ దెర్ ఇస్ నో బిగ్ సైన్స్ హియర్ ఐ యామ్ యూటిలైజింగ్ ఆల్ ద waste samples to convert into energy. Almost 7-8 years back I thought I want to use the meat waste. Now the price of meat is more than 900 or 700, something like that. So you can't get the waste from that. So I decided to go to chicken waste. Even chicken prices are, fluctuating. Like in one season 250 and another season 150. So I want to use this somehow animal waste, I can say honestly. If you go to any seashore, you can see a lot of fish waste, like head type something. I really don't know much about the parts of the fish, but waste is, you can see waste in several places. So I want to use this all the ways, like you can say that algae and then fish waste or any animal waste. So in that case, how to generate the energy? Before going to start my presentation, so what is energy? Especially not for this slide is not for faculty. This slide for the students. There are 3 kinds of science still in the society. There are 3 kinds of science. One is atmospheric science. If you say atmospheric science means, don't see the physics people. Even biological people can do the atmospheric science. So there is life. That's why NASA is always top in the science. There are 3 kinds of science, you can say. One is atmospheric science, other thing is public health. We already faced the COVID, and we don't know what other COVID will come tomorrow. So public health is one of the other major area to do science. This is for your information. And third one is the energy. Because agriculture, we do have a lot of varieties of rice, wheat. That's why funding is a bit shaky than other, compared to these three areas where I'm telling this atmospheric science, public health as well as energy. So before going to start this energy will dictate the economy of the country. Means, very recently, last before a year, Sri Lanka faced severe economy due to only fuel problems. Majorly, 1 liter of petrol 5,000 in Sri Lanka just 2 years back. If you think that if there is no petrol, diesel today, at least for next 10 days means you won't come to class. And nothing will be transported, including tomato, rice and all from the fields to market. Even after 10 days, you will severely, affected because of this energy lacuna. So that is the case, and I just before I said that continuous increase of continuous increasing this petrol or diesel prices. Alarming the situation. Why this situation happening? Why this much hike? Why this much hike? So that is too much in the process. So let it be like this only. Sir, off chasing ajayali. So let it be. So energy, whatever, it dictates our life also, and the energy and will dictate the economy of the country also. So ఈ ఎకానమీని మనం ఎట్లా కంట్రోల్ చేసుకోవాలంటే వన్ ఆఫ్ ద ఒకప్పుడు అగ్రికల్చర్ ఫ్యాక్టర్ ఉండిందండి, అండ్ వీ డూ హ్యావ్ లాట్ ఆఫ్ రైస్ వెరైటీస్, వీ డూ హ్యావ్ లాట్ ఆఫ్ టొమాటో వెరైటీస్, అండ్ వీ డోంట్ నీడ్ టు స్పెండ్ మచ్ మనీ, వీ జస్ట్ ప్రొటెక్ట్ దెమ్. ఉన్న రైస్ని ఉన్న టొమాటోని ప్రొటెక్ట్ చేసుకుంటే చాలు. సో డజంట్ మీన్ దట్ ఐఎమ్ నాట్ డిస్కరేజింగ్ అగ్రికల్చర్ సైన్స్. వీ నీడ్ టు గో ఫర్ ఎప్పటికప్పుడు న్యూ వెరైటీస్ తెస్తూనే ఉండాలా, బికాస్ క్లైమేట్ చేంజ్ ఇస్ ఆల్వేస్ రెసిలియన్స్, కొంచెం ఇట్ విల్ బి ఆల్వేస్ షేకీ. సో వీ నీడ్ టు ఫోకస్ ఆన్ ఈవెన్ అగ్రికల్చర్, బట్ ఐఎమ్ టెలింగ్ దట్ we have to focus on these three areas which I already mentioned. So my area is one of the areas, energy area, where you can focus. And other thing secret ఏంటంటే ఎక్కువ ఇంపాక్ట్ పబ్లికేషన్స్ వస్తాయని అది చెప్పుకోకుండా మీరు అన్ని చాలా ఫాస్ట్గా చదువుకుంటాం. అది ఇదేనే ఎవ్రీ సైన్స్ హ్యాస్ ఇట్స్ ఓన్ వాల్యూస్, ప్రతి దానికి ఉంటుంది, దాన్ని మనం తప్పపడాల్సిన అవసరం లేదు. At the same time, there are two kinds of energy, renewable and non-renewable energy. This slide mostly for the students, not for the faculty. So renewable and non-renewable energy. What is renewable energy? What is non-renewable energy? Whatever we are using right now is the non-renewable. Non-renewable means you cannot get back in a short time. It takes more than 300 and 600 years. All the hydrocarbons, that includes whatever the fall in the trees, leaves, and yourself and bacteria and all, go deep in the earth crust mantle, convert it into crude oil for more than 300 years it will take to convert into crude oil. That crude oil we need to take and purify it, and then and we can get the petrol, diesel, kerosene and all. That is the almost like 600 years story. That's why I am telling that non-renewable energy means you cannot get back in a short time. You cannot get back in a short time. You need to focus on at least 600 years. We need to wait for the, once if you put a petrol 1 liter in your bike and go around that town and come back, it will exhaust. You can't get back the petrol. But with the case of renewable, I will come back. Renewable means very frequently you will get the energy from renewable sources. That includes the solar energy. Every 12 hours you can see the sun, even if it is a summer or winter, any season, right? So this 12 years, within every 12 years you can see the sun, and at the same time, every time you have a wind energy sources. You can see the Galimadal అంటారు తెలుసు కదా కొండల మీద పెట్టుకుంటారు. Wind energy. So you can see that and all you can get in a cheap way. Solar energy, wind, tidal and waste, bioenergy. So this and all you can get back, but the problem here is people are always trying to say that how best these renewable energies implemented in India. People are telling always solar energy, wind energy, bioenergy, tidal energy, geothermal energy, hydro energy. This is all renewable, but how best it is implemented in India as per the statistics. I'll tell you some of the stories, but till date we are using more than 70% non-renewable fuels only. ఇప్పటి వరకు non-renewable fuels ఏ మనం వాడుతున్నాము. But we are telling about the always renewable energy. So this is the types of energy. This is just for this slide for the students. So you have a renewable energy, solar, hydro, wind, tidal, biomass, geothermal. That includes this particular renewable energy resource is very focused area. Though we depend on the other side, non-renewable more than 70, but we need to think of the alternate sources, because regularly every day we are using all the petrol diesel sources. Every day it is depleting all the sources. That's why we need to focus on. That's what I said very beginning slide. ఒక దేశం యొక్క ఆర్థిక పరిస్థితిని ఒకప్పుడు అగ్రికల్చర్ సెక్టర్ డిసైడ్ చేస్తే ఈ రోజు ఎకానమీ ఎనర్జీ సెక్టర్ కూడా డిసైడ్ చేస్తుంది. That's what I said, that just think of that today there is no petrol or diesel, and think of next 10 days if there is no petrol or diesel, what is your situation? And you don't come to classes also. Just forget about the market and all or logistic portions. So and then that's what I said, people are always thinking the renewable energy in India, how best you are using. Always telling, I myself, one professor asked me in BIT value presentation, you're always telling the biodiesel from fish waste, how much is using now? It's zero. Still we are working on that. Now, this is the energy consumption in India: coal 44%, and then petroleum 25%, and then natural gas 6%, and electricity 3%, and renewable only 22%. At least we are in a stage to show 22% in India. In 2020-21 means every year if you go MNRE, Minister of New Renewable Energy Sources, and see the statistics where this number is 22 is increasing every day. Every day it is increasing. At least we have to reach more than 50 and just forget about the fossil fuels. Fossil fuels means non-renewable energy. Petrol, diesel and all purified from the crude oils. Why we are suffering? Why we are suffering with the US now? Only one reason. Students, only students. Why we are suffering with US now? Students only, please. ఎందుకు మనం సున్నం పెట్టుకుంటున్నాం అమెరికా వాడుతాం? Yes, వాళ్ళ దగ్గరే కొనాలనేది వాళ్ళ అంతేగా. ఆ సోర్సెస్ మనకే ఉన్నాయి ఈరోజు ఆ సోర్సెస్. అసలు ఇందాక చెప్తున్నా వాట్ ఎవర్ ది సోలార్, విండ్, సీరియస్లీ focus on the solar and the wind energy. We don't need it. We don't need any fuel, any raw crude oil from any sources. We do have already. So before going to start this, this is the story of renewable energy, statistical story in India. So biofuels, my portion is just one is solar energy, wind energy, tidal energy, bioenergy, hydro energy, and geothermal energy. Bioenergy is one of the which is a part of my research, but how best it is implemented? Can anyone answer? Everybody is asking. You are telling bioenergy. How much is got in? Like if you go to HP petrol pump, Bharat petrol, you will see the non-renewable fuels. Where you are getting bioenergy? Any one example? Anybody? it is implementing seriously. One biofuel. There are three kinds of biofuels. One is bioethanol, biodiesel, and biohydrogen. Biodiesel is a buzzword now. Surely we will depend on the hydrogen only in near future because all these. Any one thing you tell me, which one is very old supplement of the fuel?
Biogas.
Yes. Everybody forget about the bioethanol. It's a old story, more than 500, 600 years back. People generate the ethanol. What is the use of ethanol? Not only for the alcoholic consumption, it is the supplementary for the petrol or diesel. Say for example, what petrol diesel contains? Hydrocarbons and impurities like lead and all. If you burn one liter of petrol, say for example 1 liter of petrol will burn means 100% of pollution means if you blend with this ethanol 10%, E10 that is nothing but. That means దొంగసారి రాస్తా ఉంటారు తెలుసు కదా దాని గురించి ఎక్కువ చెప్పుకోవాల్సిన అవసరం లేదు చాలా చీప్గా కూడా చేయొచ్చు బై టేకింగ్ ఎనీథింగ్ మచ్. So if you blend this 10% ethanol with 90% petrol or diesel, whatever it may be, petroleum products, and then you will reduce the cost as well as the pollution. Now the job is in the hands of engineering department. We have engines E10, E50. That means E10 means blending with 10% ethanol, and we need to make up that story into 50%. That means 50% petrol and 50% ethanol. So for that, the people are trying always E10 vehicles, and we need to go with the E50 also. Now, so before going to start these biofuels, okay. So I'm going to use this bioethanol. What is the major source of ethanol? ఇథనాల్ అనేది ఎక్కువ ఎక్కడి నుంచి వస్తుంది? The US and Brazil are the two top countries to generate the ethanol. Surprisingly, one is in corn, other one is the sugar cane. And you can get from even rice also. So corn and sugar cane are the topmost sources for the ethanol production in US and Brazil. US is the top and Brazil is, both US and Brazil. One is depends on the corn, another one is depends on the sugar cane. If you generate this ethanol very frequently, you can reduce the pollution also. That is the ethanol concept. I'll come back after some time with the ethanol story. I'll first go to the buzzword like hydrogen. What is hydrogen? How can you get the hydrogen? There are three concepts. I will also come later, but these samples, whatever you are using, the waste sample, just I mentioned the algae and fish waste, you can get anywhere. That's what even మనం కొంతవరకు అసహ్యంగా చూస్తాము పాచిని. Nothing wrong in that. Even if you go to Japan and southern eastern states, eastern countries, they will eat like మనం ఎట్లా కరవాడ తింటామో ఆల్గేని బాగా రిఫైన్ చేసుకొని ఎండబెట్టి మనం ఎట్లా ఈ పికిల్ తింటామో ఆ విధంగా తింటారు వాళ్ళు పాచి. ఏం తప్పే ఉంది, ఫోటోసింథటిక్ ప్రోడక్టే కదా అది కూడా, నథింగ్ రాంగ్. ఆ మురికి నీళ్ళలో పెరిగే చేప తినే నువ్వు ఆ పాచిని ఎందుకు తినవు అనేది కాన్సెప్ట్ చాలు. And nothing wrong in that also. So algae is majorly divided into three groups: micro, single cell, as well as the multicellular. It ranges from the single cell to big plants. So various why people are choosing algae means you can handle very easily in the experiments because it's a single cell. Whatever alteration you want, you can do. Whatever genetic modification you want to do, it's a simple life cycle, as well as the efficient photosynthetic organism. Efficient photosynthetic organism. Even ekkada tadi unna grow అయిపోతాయి. Even it grows in a potable water also. That's why people always prefer algae for the Any products, don't think that bioethanol and biohydrogen or biodiesel for the algae. Algae is one of the best nutraceuticals. Medicine people, I mean, medical side research people will always prefer this algae to get the secondary metabolites like alkaloids, terpenoids, phenol, something like that. What depends on the algae. It is one of the best nutraceutical source also. Now, before going to start my algae story, I used to generate this biohydrogen and bioethanol from algae. So how to get the algae? A single cell algae in vitro inoculation, and just go with the anaxic condition. Anaxic condition, I will tell you. I'll show you. Then get the hydrogen gas. Simply, this is a very old story. Get the sugars, either it may be grapes or either sugar cane, or just get the sugars. Add some yeast. You will get the alcohol in the process of fermentation, which is under anaerobic conditions. Kinva prakriya under Telugu. You will get the alcohol with generally yeast. Even some bacteria will also works for the first to generate the ethanol. So coming to main topic, biohydrogen. Why people are preferring biohydrogen? After some time, you will definitely forget about the petrol, diesel, and all. Everything depends on the biohydrogen. There is a train in Germany now. The train name is Hydride. Very recently, 1 month back, ఒక ship the hydrogen as an energy source. Hydrogen bomb విన్నారు కదా. It once ఒక్క hydrogen split అయితే ఎన్ని కిలోల gasoline 30,000 calories అనేది ఎనర్జీ which is not equal to gasoline, more than gasoline. And then say for example, here I want to tell you one story. How you get this hydrogen from the plants or algae. Other side of the coin is this is 3/4 of the water. This contains 3/4 of the water and 1/4 of the land. That means 3/4 of the electron in the H2O only. H2O. All chemistry, material science, nanotechnology, these people are trying to get hydrogen from the H2O through catalysis, electrocatalysis, using the nanoparticles. Always they are trying to split the water. Always they are trying to split the water with the help of catalyst. By splitting the water, they will get the hydrogen. Because I already told you this entire earth contains 3/4 of hydrogen is there in the form of H2O more, but in the atmosphere it is 0.01 only. You imagine and see the statistics. Always chemists and material science, nanotechnology people will try to split the water to get the hydrogen. With the case of plants, there is a mechanism in this is nothing but water splitting or Gill reaction. That's a God's gift. Plant itself split the water. Not only plant, that includes the algae. Split the water to give the hydrogen. Protons. I'll tell you that story. How this hydrogen will come. Say for example, algae. Specifically, we know very since beginning. Childhood, like Chlamydomonas, right? Which have, this Chlamydomonas. If you consider this, how it photosynthesizes. How you get this hydrogen means, if light is there, normally it goes regularly to the, water splitting, electron transport chain, the movement of electrons, and it goes to the fixation of Cp2 in the process of process of CPD cycle, Kelvin-Bentham-Marshall cycle, simply Kelvin cycle. CO2 fix here, and finally you get the glucose C6H12O6. That is a regular phenomenon. If you get, that is nothing but photosynthesis. 6CO2 plus H2O in the presence of sunlight, you get the C6H12O6 with the result of O2 also. Why? That's why people are always telling why you do plantation means you will get the O2, but you need to think that CO2 will also consume. One of the biggest greenhouse gas will consume in the process of photosynthesis. మనం మొదలే CO2 ని మనం, please remember this, CO2 plus H2O in the presence of sunlight, you will get the carbohydrate that is required photosynthesis. That tells some story. What other story means when cell, chlamydomonas or any algal cell, if it won't because of too much light and it's sufficient for photosynthesis, always it goes to under some stress condition. Say for example, more CO2 outside, more temperature, or more light source, or more water also. In that case, this cell always goes to alternate pathway. The switch of the photosynthesis, and whatever generate this H plus protons, this converted into hydrogen in the presence of hydrogenase. This is an alternate pathway, and entire photosynthesis people, they forget what will happen once water split. What is the fate of this? Always people are thinking that water will split and get the oxygen in the process of photosynthesis, and nobody will think about what will happen these protons, 4H protons. Once water splits in the process of Hill reaction, what will happen to these protons, 4H plus or H plus, whatever it may be. Where it goes, these H plus protons? It goes and converted into hydrogen with the help of hydrogenase and this electron transport system, whatever you have the electrons. This story never knows. This story never knows. Anybody know? Everybody know about the photosynthesis. Everybody know about water splitting. Everybody know about Hill reaction. Everybody know about the formation of, fixation of CO2 and formation of C6H12O6. But nobody knows what will happen to these 4H. Not only here, 4H plus. There are some pathways, H plus protons, where it goes finally. It will remain as a fermented product once if it kills and go with the fermentation. It is also one of the fermented product, but nobody knows that what will happen to this 4H. This is nothing but, that's what I said, God gift. We can use that 4H. Actually, we can't use, plant will use that 4H and get the hydrogen with the help of hydrogens. Only thing is, we need to stop the chlamydomonas, stop your photosynthesis, go with the other. That's what we need to do. Just give some stress to them and get the hydrogen. Either it may be give more CO2, or more than sufficient light and more than sufficient temperature, or simply just lack of water. It goes to the alternate pathway, that is nothing but generation of hydrogen. And this story almost very recent story, 10-15 years back story. More than 200 years people know about the photosynthesis and nobody knows about this. More than 200 years people know about the photosynthesis. What will happen to that H plus? That's what I said, the God gift. You can get the hydrogen through conversion of this hydrogen into hydrogen gas with the help of electrons which generated in the electron transport system. So that's the story, whole story of getting hydrogen from the algae. ఎట్లా వస్తుందో అర్థమైందా? ఫోటోసింథసిస్ తెలుసా? How you get the hydrogen? At least one word. By seeing that. I'm very happy if you understand this process. Everybody know about the biodiesel, everybody about the bioethanol. Just take some sugar and keep some fermentation, you get the ethanol, that is no. But I want you people have to know that this hydrogen will come this way. At least one people, if you understand, I'm very happy. This is the only crucial part in my talk. Only crucial part. Everything, other things, everybody know. I want to tell you again and again and again. This part is very crucial in my talk, and even if you don't understand other things also, it doesn't matter to me. If you understand this point, what will happen to this H plus protons? Okay, that is only give the hydrogen, which is considered as the future. The future clean energy is only hydrogen, because I said 30,000 calories of energy will come. 120 kilojoule, 122 kilojoules per gram of any biomass you will get the hydrogen. That's the actual story of this hydrogen. And then there are 3 steps how you can generate. That means we need to enhance the photolysis of water or splitting the water. It's nothing but the Hill reaction, nothing but the splitting of water. We need to enhance this reaction to get more hydrogen, to get more hydrogen. At the same time, oxygen always inhibits the hydrogenase. You need to think of hydrogen is insensitive to sensitive to oxygen. That means it cannot tolerate. Hydrogenase function will only under anaerobic condition, under completely not even hypoxia, anoxia condition only. Anoxia means complete absence of oxygen. That means anaerobic conditions. If oxygen is there, even if it is a hypoxia, even a small amount of oxygen, this hydrogen gene cannot work further. So only thing is only way we need to stop the photosynthesis. If photosynthesis continues, hydrogen cannot produce, because photosynthetically produced this oxygen will inhibit this function. That means we need to completely avoid, damage the photosynthesis. We cannot remove it. We can damage our insist or hold on for a while by giving some stress. In that case, we will get the hydrogen. Otherwise, you won't get. That's why I said the word alternate pathway under stress. Some people won't believe this theory, but this theory may be good or bad just 7-8 years back. But the question finally, people are thinking, what will happen to this express? That is whether this works or not, I'm not sure. But it is working now. 70% of hydrogen will through this pathway only. If you just crush all the algae and give fermentation for. Previously, we are seeing hydrogen only with as a fermented product, sulfur, hydrogen, all ethanol and all as a fermented product. But now this is a product of alternate pathway in photosynthesis. So that is the story. So there are 3 steps, and the other key factor is efficiency of hydrogenase. One algae contain 1 hydrogenase, another algae contain 2-3 copies of hydrogenase gene, and other maybe 6-5. That's why each algae has its own specificity. Chlamydomonas will respond to the some extent, and even it is a model alga, it responds in some way, and some hyperactive algae is there, it will respond more. Hypoactive also there, some are response very less when compared to model alga, Chlamydomonas. This is a story about how best we can generate the hydrogen from the algae. So I want to tell this role of nanopart. This is almost 10-year story. Finally, we got some conclusion. So this is the way my student who died in the COVID when he was their postdoc in China, Dr. Paramesh. So postdoc long. That's why I want to remember this. He is the man who worked on hydrogen from algae. The first man died in Chandra. So he's the man behind of this work. So how you get the energy? So just inoculate the algae and then culture it. Go with take a biomass, equal biomass of all the cultures, and make an estimation of biomass and give some stress and estimate that stress may be either the temperature or pH alteration or whatever the stress, or add some nanoparticles or remove some genes and all, any kind of stress you can give and get the hydrogen data from this stressed one compared to control. So that is the story of getting hydrogen from the algae. Easy story. So now my university has one man, Professor Sekhar. Last time he came here, he is the person who worked on TiO2, titanium dioxide. He always worked on titanium dioxide with a lot of Google citations. So what is why I am choosing this TiO2 for my work is the basic function of TiO2. Of course, the people are used to some medical side and other people they use for many things also. TiO2 is the basic function of the TiO2 is it splits the water. It acts as a catalyst. That's what I showed just before. Chemistry people, nanotechnology, material science and all, they are always trying to split the water to get the hydrogen. But biology people always trying to add some catalyst to enhance the photosynthesis pathway, like especially photolysis of water, splitting of water. That's why I choose this TiO2 to enhance the Hill reaction, to enhance the splitting of water, to enhance the photolysis of water process. So that's why I choose this TiO2. And then unfortunately we got good results with the short treatment. Say for example it works only for 12 hours. After that control is getting more hydrogen. That means though I choose this catalyst for water splitting, until 12 hours it works, and it works well for the splitting of water, we are getting more hydrogen from the for 12 hours. After 12 hours, around one day, we giving toxic to the algae. Toxic to the algae. So selection of the catalyst also the important factor for this. So here we made some of the nano TiO2. Generally this is a nano particles, but I am making some nano rods, nano pores. There are some difference the results between nano particles. So we are working on that to get them more. But absolutely we got good results with this. We need to work on more. And I told you oxygen is the inhibiting factor for hydrogenase. If oxygen is there means hydrogen cannot produce because hydrogenase is sensitive to oxy conditions. That's why we need to create an oxy condition, not even a hypoxia. Hypoxia also gives on. So under this condition we got a good results around, let's say for example, we are by adding some oxygen and sodium bisulfate is NaHSO3 is a sodium bisulfate. It's an oxygen scavenger which scavenges oxygen whenever the oxygen produced. If any possibility is there in the algae, in that case it will absorb the scavenger. That's why we added TiO2 and oxygen scavenger. It worked very well for us. This is a different story. Why I keep this algae means algae growth assay. That's what I said. Each and every algae has its own potential. We do have a different metabolic rate. Some person has this much of rate and other person has this much rate. So algae has its own limitations. Certain algae will respond more, some certain algae will not respond. See here, you can see, always we feel this Chlamydomonas reinhardt is a model algae. It will respond for many and genome sequence, everything is done with this and we do have a lot of experiments, a number of more than 10,000 papers are there in Chlamydomonas. This is only my vision, 10,000, more than us. So, some algae like Astrasis quadricellular and Chlorocopamine and some of the green algae, the same family, even under dark also it is giving biomass. So how can I say? How can I stop? Even if you say this, you can see this Astrasis quadricellular, a unicellular green algae, almost like Chlamydomonas, it gives growth under dark condition. Even if you throw anywhere pH condition, even if in acidic condition, it will grow. That is why I am calling this one and Chlorocopamine 2,3 algae. In my lab, I have 2 hyperactive algae. Even any condition, it will grow. Even if you add until 3 acidic pH condition, 3 means it's a serious. Acidic condition 3 pH means it's Mn is required for as a trace element. And then it has both, it is a core part of some photosynthetic activities in oxygen, ROS, scavenging process, and CO2 fixation also, Kelvin cycle also. If it is there in the form of MnCl2, always the character is more positive. It's there in the oxides of manganese. That means it has even negative point. Oxides of any element is negative point. At least worst case we have some. So that's why I published. This is the first report on this. MnOD is involved in carbohydrate and nitrate metabolic pathways, and it involves in the ROS scavenging. It involved in CO2 fixation. What is CO2 fixation? ఏమంటారు pathway? అప్పుడప్పుడు అడుగుతున్నాను. CO2 fixation pathway ఏమంటారు? Krebs cycle ఎక్కడ వస్తుంది? పొడిసిస్లో వస్తుందా? Kelvin cycle విన్నారా? CPP cycle ఇందాకే చెప్పాను. Kelvin cycle, Kelvin Bentham Hashem CPP cycle, where you will get ఏదైతే వదిలిస్తున్నావో CO2, it got fixed in that cycle, Kelvin cycle. So this MNO has different roles, and we have excellent. We did this MNO nanoparticle and analyzed with the help of SEM, MIDAX, and XRD. So we got very good, accurate, successful synthesis of high quality MNO to nanorods with homogeneous morphology and well defined knowledge. It's a different story. People are always trying to use nanoparticle. I want to tell you one word because మీకు అది కొంచెం చెక్కి చేసి వెళ్ళిపోతాను, ఎక్కువ అవసరం. What is nanoparticle? ఓకే వాట్ ఈస్ ద స్పెషల్ క్యారెక్టర్ మీన్స్ వాట్ ఎవర్ సింపుల్గా చెప్తాను దాన్ని. కలకండ తీసుకుంటారా, ఒక గ్లాస్లో వేయండి కలకండని బాగా చిన్న పొడిగా చేసి వేయండి. ఏది ముందు కలుగుతుంది? ఎందుకు? రియాక్టివిటీస్ మోర్, సర్ఫేస్ ఏరియా తక్కువ ఉంటుంది, రియాక్టివిటీ మోర్ ఉంటుంది. ఈవెన్ అది విషమైనా నువ్వు ఒక ఈ ఏదైనా టాబ్లెట్ ఫామ్ ఇస్తే ఒక 2 minutes చనిపోతే దాన్ని పొడి చేసి ఇస్తే 1 minute చనిపోతాం. బికాస్ ద సర్ఫేస్ ఏరియా వెరీ లెస్, యూనిక్ బయోఫిజికల్ క్యారెక్టర్స్ అండ్ రియాక్షన్ ఈస్ వెరీ more even if it is a poisonous. Okay, so that's why I want to tell you that we successfully synthesized this amendment to for the algal cultures. So we got a excellent biomass at 0.1 nano mmol. So with this, after getting the biomass, we just go through. If you remember the previous slides, once if you get the growth, boil it and then boil it and spin it, remove all the debris, green debris. Take the solution, add a beta alpha malase, and then harvest the carbohydrate solution, add yeast. That's all. It will automatically get it because sugars are. If yeast will always prefer the sugars and it converted into ethanol. That's the story. Very simple story. We got 30.37 grams per liter of ethanol. With the feedstock grown in a tap with 0.1 mg of nano. That means 0.1 mg of nano mmol gives more biomass. ఆ కాన్సంట్రేషన్ అనేది ఎక్కువగా ఆల్కలో బయోమాస్ ఇచ్చింది. ఎక్కువ బయోమాస్ ఉందంటే ఎక్కువ షుగర్స్ ఉంటాయి, ఎక్కువ షుగర్స్ ఉందంటే ఎక్కువ ఇథనాల్ ఎక్స్పెక్ట్ చేయొచ్చు. ఓకే, దిస్ హోల్ స్టోరీ డిపెండ్స్ ఆన్ 0.1 mg per liter nano mmol works very well. That's all, the only single line for the whole paper. So this is, I wanted the possible role of MnO2 and biomass and ethanol production. It majorly here water splitting lo untundi, major, and then RS scamming lo untundi. If any stress condition vachina through SOD it works, and then it is involved in CO2 fixation in Kelvin cycle. So certain role, prati daniki oka role untundi. Why the biomass is enhanced, why the ethanol is produced. So this is the way you can. కొంచెం బోర్ కొడుతుందేమో, కొన్ని చూపించకూడదు, కొన్ని చూపించకుండా వచ్చి పాఠం చెప్పకూడదు. ఈ పాత్వే అంతా చెప్తున్నాడు, ఇవన్నీ నాకు అనుకోవద్దు. ఆడికి వస్తే ఇంకా నేర్చుకోవాల్సి వస్తుంది. కొన్ని బేసిక్స్ చెప్పాలి నేను, కొన్ని అడ్వాన్స్ కూడా చెప్పాలి. దాన్ని ఎందుకు లేదు మనకి ఇవన్నీ అనుకుంటే నాకు కూడా ఎందుకు లేదు. ఎనీవే, బయోడీజల్ ప్రొడక్షన్, very beginning I told you that fish is one of the best source. At the same time, you cannot buy the fish, right? We are using only fish waste, where if you go to entire coastal zones, lot of fish waste is there in the seashore. Early morning నువ్వు వెళ్ళామంటే ఒక సంచులు వేసుకోవచ్చు, ఫిష్ వేస్ట్ ఒక సంచుల్లో వేసుకోవచ్చు. One way you are converting that fish waste into energy, other way you are cleaning that area, waste management practice. Here I want to tell you one thing. One IT professor in VAT presentation, she asked me, you are collecting all the fish waste, crush చేసి బయో ఆయిల్ తీసి then go for the transesterification to convert it into biodiesel. అంత నెసెసరీ ఏముంది? ఆముదం తీసుకో, గ్రౌండ్నట్ ఆయిల్ తీసుకో, వెజిటేబుల్ ఆయిల్ తీసుకో, లేదంటే ఫిష్ ఆయిలే తీసుకో, డైరెక్ట్గా చేయొచ్చు కదా. ఎందుకు నువ్వు దాన్ని ఎక్కువ చెప్తున్నాను? ఫిష్ వేస్ట్ తీసుకున్నాను, బీచ్ నుంచి తీసుకున్నాను, చాలా తిరిగినాను, అదంతా తిరిగి తీసుకున్నాను, నేను ఎందుకు చెప్తావు అంటే, ఆముదం ఆయిల్ తీసుకో, లేదా గ్రౌండ్నట్ తీసుకో, ఎనీ వెజిటేబుల్ ఆయిల్ తీసుకో, కన్వర్ట్ అని చెప్పినాను. మేడం యూ మిస్ ది వన్ పాయింట్. నేను అదంతా ఆ స్టోరీ నేను నడిచిందంతా ఎందుకు చూపించానంటే ఆ ఫోటోలు. కన్వర్టింగ్ ద వేస్ట్ అనేది వన్ ఆఫ్ ద బిగ్గెస్ట్ వర్క్. వన్ ఆఫ్ ద దట్స్ వాట్ ఐ సెడ్. ఐ ఆల్వేస్, నేను వచ్చిన చాలాసార్లు ఇప్పుడు DBT పదం వాడాను. DBT ఫండింగ్ ఇస్తుందంటే గ్రేట్గా ఫీల్ అవ్వాలి. యూ నీడ్ టు యూజ్ దిస్ ఆపర్చునిటీ. నియర్ ఫ్యూచర్ దేర్ ఈస్ నో ఫండింగ్ ఫర్ ఈవెన్ ఫర్ సైన్స్ ఆల్సో. What will you do if there is no finding? You need to depend on the cheap sources like fish waste. నాకు వచ్చిన అనుభవంతో ఎక్కువ గ్రాంట్స్ రాదు అనుకున్నప్పుడు, take the waste and convert it to grant and automatically to give good papers also. There you need to guess the point. not necessarily. And I will tell you one more story here also. మనం ఇంట్లో వంట చేస్తాం, ఆయిల్ తీసుకుంటాం, అందాలు ఎన్నిసార్లు చేస్తాం, వడలు ఒకే దాంట్లో 1-2 సార్లు చేస్తాం, లేదంటే కూరకు వేస్తాం. ఆల్ పెద్ద పెద్ద ఇండస్ట్రీలో హోటల్స్లో చేసినట్టు యూ కెన్ కలెక్ట్ ఆల్ ద వేస్ట్ ఆయిల్ and send it to the biodiesel industry. They will convert that. That is also possible. Not necessarily you don't need to depend on the fish waste. So as I said, the fish is an oily one and it is a source for PUFA, polyunsaturated fatty acids, one of the biggest protein source. Fish meat is one of the best when compared to chicken and other animal food sources. Schematic representative from biodiesel production from fish waste. You can collect from all the fish tails. Fish అంటే ఫిష్ లాగా ఏం పడుతుంది అక్కడ, టైళ్ళు, కాళ్ళు, చేతులు, ముక్కులు, తల, ఏదో ముక్కులు ఏవో దొరుకుతాయి, అన్ని కలెక్ట్ చేసుకొని సెపరేట్ ఇట్ అండ్ దెన్ treat it, acid treatment, treat it because it got old, you don't know anything, treat it, depends on the, first segregate it, divide the tails, divide the heads, divide the remaining portions, separate it, segregate and separate it and even if possible just identify that what is that fish. Is it a very useful fish or even fishes or poisonous fish also there. So collect that fish waste, segregate it and separate it and isolate it and then go with the treat with it to get the fish oil. దీనికి పెద్దగా సైన్స్ అవసరం లేదు. బయటకు వెళ్ళేసి రైతు బజార్లో ఒక ఫిష్ తీసుకొచ్చి ఎండలో పెట్టేసి బాగా కట్ చేసి ఆటోమేటిక్గా ఆయిల్ బయట వస్తుంది. అంతెందుకు ఫిష్ మనం చేసేటప్పుడు పెనం మీద పెట్టండి, ఆయిల్ ఆటోమేటిక్గా వస్తుంది. యూ డోంట్ నీడ్ టు క్రష్ ఎనీథింగ్, ఏం చేయాల్సిన అవసరం లేదు. సో టేక్ దిస్ ఫిష్ వేస్ట్, బాయిల్ ఇట్. Muffle furnace లో 500 degree లో పెట్టేసినామంటే you don't need to use many solvents also, just use water to get the oils from the fish. Just use water to get the oil. No need nothing. Just take a distilled water, add this fish waste in muffle furnace 500 degrees centigrade, it will be automatically. And I'm not telling the exact the oil, kind of oil. Later you can separate it and then from that oil you can go with the production of what is diesel, ETH and ETOH and ethanol diesel, H2 and hide. What is biodiesel? Fatty acid methyl esters or ethyl esters, alkyl esters, nothing more. Ester equation is trans esterification జరిగినప్పుడు either it may be ethanol తో కలిపితే మనకు వచ్చే ఆ ఫేమ్ or fatty acid methyl esters డీజిల్ అంటే దాని డీజిల్ అండి. వెరీ సింపుల్, దాన్ని చాలా పెద్దగా. దిస్ ఇస్ ద ప్రోటోకాల్. వీ కెన్ టేక్ ఆయిల్, యాడ్ మిథనాల్, నాట్ నెసెసరీలీ టేక్ ద లైపేజ్, అండ్ దిస్ ఇస్ ద ప్రొసీజర్ యూ కెన్ catalyze it and get the FAME or fatty acid methyl esters. Nothing but diesel like. Whatever you get the diesel like, you compare with the diesel by this one the why it is not changing. You could shoot them. Stop the way. Very cheap source. This I can say collection of fish waste. Another student from Pile, Rajendra, he is working on chicken waste. And initially he started with meat waste. Fortunately, meat crashed 800 or 900 rupees. He just stopped work on stop collecting the waste because there is no waste. No problem. So this is why you can use the waste to get the products. నేనేదో బయోఇథనాల్, బయోడీజిల్, బయోహైడ్రోజన్ చెప్పాను అనుకోవద్దు. ఎనీ వేస్ట్ కెన్, ఎనీ బయాలజికల్ ఆర్గానిజం వేస్ట్ హ్యాస్ సమ్ వాల్యూ. బచ్చలాగా తీసుకున్న సమ్ ఆల్కలాయిడ్స్ లాంటివి వస్తాయి, ఆ వేస్ట్ నుంచి కూడా ఏమైనా చేసుకోవచ్చు. గో విత్ దట్. చిన్నప్పుడు RRR అని, ఇప్పుడు 4R బీయింగ్ యూజ్ రీసైకిల్, లేదంటే 3R అని, 4R అని, 6R అని, ఏదో R చెప్తున్నాను. R will always increasing. కొత్తగా ఒక R తయారైంది. సో క్వశ్చన్ మాత్రం అట్లే ఉంటుంది. ఇటు 3R ఇచ్చాడు, 4R పెట్టుకుంటాడు అక్కడ వన్ ఆఫ్ ద యూనివర్సిటీస్లో వచ్చింది. సో reuse it. Anything you can do is waste. Simply this talk is production of commercial products through waste, that's all. ఎట్లా నువ్వు దీన్ని చేసుకుంటావు commercial products, not only this one. Just now I said that one of the dental doctor, he isolated chitosan from biopolymer from honeybees waste. Honeybee rectal నుంచి పడిపోతుంది ఏపీ, అది జస్ట్ చాలా లైఫ్ టైం తక్కువ. honeybee waste నుంచి he extracted the chitosan biopolymer for dental activities, of course biological. So this is one of my students' data. He did some of the bio oils and sent to the GCMS Mahila University. We have 7 fatty acid methyl esters. 87.2% of this oil, biodiesel-like oil, is matched with the original diesel. మనం పెట్రోల్ బంక్లో నుంచి డీజిల్ తీసుకొని మాది ఇస్తే 87 పాయింట్, అంటే యాక్చువల్ అయితే 95 దాటాలంట, 97 దాటితేనే ఒరిజినల్. సో అది అయిన తర్వాత అనదర్, బట్ ఫార్చునేట్లీ మీట్ వేస్ట్తో చేస్తే 92% వచ్చిందంట. ఈజీగా చెప్పొచ్చు అక్కడ లాజిక్ అంటే మీట్లో ఎక్కువ ఫ్యాటీ యాసిడ్స్, సో ఈజీగా వచ్చింది. ఈ స్టోరీ ఇంతకు ఆపేశాను. అండ్ దెన్ so now if you go to any fish industry or aquaculture sectors లో వెళ్తే దాన్ని చక్కగా మేబీ కొట్టి అన్ని పాసం చేస్తాం. మిగిలిపోయిన వేస్ట్ యూ కెన్ యూజ్ ఇన్ బై టేకింగ్ ది aquaculture, or you can see this kind of situations in the seashores. Either way you can get the fish waste to go for the biodiesel production. And we recently focused, one of my student is working on this in algae. Algae కూడా ఫ్యాటీ యాసిడ్స్ ఉన్నాయి, బట్ నాట్ ఆల్గా. Algae is most famous for carbohydrate because photosynthetic organism, but one student is working on algal fatty acid pathway. Is there any knock the genes like fatty acid fatties? Is there any, if you do something, she is working on that, it's not yet completed. She is working on that to knock the genes of certain genes related to fatty acid metabolism to get more fatty acids. In that way you can get the more diesel. That is the work, and it's not completed yet. Conclusion: the whole algal samples are best biological sample for biodiesel, bioethanol, including biodiesel, but only these two is famous now due to simple life cycle and efficient photosynthetic capacity. Our laboratory also established the augmented production of biodiesel from fish waste, and still effective protocols are we are developing even from the algal samples. This is the publication. The first one is the very recent January publication. So January 2026 publication. This is some of the best 10 publications. We get different papers, very recent publications for us in my laboratory. Here I want to tell you one thing. Professor Sharma అనేసి మనకు కెమిస్ట్రీలో వైవీలో ఉన్నారు. ప్రొఫెసర్ శర్మ ఆయన మిసెస్ వచ్చి ఇంజనీరింగ్ అన్నమాచార్య దాంట్లో పనిచేస్తుంది, Chandra madam అనేసి. Whatever we generated this diesel 87.2 or 92, whatever the set, we took them to we collected all the samples and we hand over the samples to the Chandra madam, and we went for the diesel engine trials until 50%, 40%, 50 is stuck, the engine is got stuck. 40% of our biodiesel and 60% of original biodiesel works very well, whatever we generated. So, ఒక చిన్న హెల్ప్ కావాలి ఇక్కడ. మేము జనరేట్ చేసి ఇంజిన్ వేస్తాం చూసారా, అవి చూపించాలి, వీడియో ఉండేది. అంటే కమర్షియలైజ్ ఎలా చేస్తారు అన్నట్టు, కమర్షియలైజ్ ఎలా చేస్తారు, వాట్ ఎవర్. అక్కడ ఒక మంచి సోర్స్ అయ్యారు మాకు అన్నమాచార్య కాలేజ్ వాళ్ళు. దే ఆర్ కన్వర్టింగ్ దిస్, దే ఆర్ గివింగ్ ఛాన్సెస్ టు యూస్ అవర్ డీజల్ లైక్ థింగ్స్ టు కమర్షియల్ ప్రాక్టీస్ కోసం అని, ఇంజిన్ ట్రయల్స్ కోసం. So engine trial test day, it works very well with until 40% blending work. Whatever we prepare the diesel. నేను కల్పన మేడం షేర్ చేస్తాను. మేబీ ఇన్ నియర్ ఫ్యూచర్ మేడం విల్ డిస్ప్లే దిస్ వన్ ఆర్ Savitri madam, ఎవరికైనా సరే. So with this, I am going to conclude my presentation. Thank you and thanks for my students, collaborators and grant agency. Very three only I got CSR, very few grants I have, nothing more than. So this is very important slide for me. For this only I came here. Thanks to everyone, but I want to promote my department. Please take a photograph with this. Okay. నాకు ఒకటి ఫోటో తీసి ఇవ్వండి. నేను ఇది కావాలి, నాకు నేను వచ్చింది దీనికోసమే. So please join in MSc Environmental Science if you have a chance. Lot of opportunity. అందరూ పేపర్ల మీద లెక్కల మీద ఆడాడ తీసుకొని చెప్తారు. మా దగ్గర చేరేది 4-5 మంది. ప్రతి ఒక్కరు మా పిల్లలకి ఎంత ఇదంటే రాత్రి CV పెట్టి తెల్లారి ఉద్యోగం కావాలంటారు. ఇక రాత్రే జరిగింది అది. So we have different sustainability. Our department always 100% employment ఉంటుంది. రాత్రి CV పెట్టాలి సార్, నాకు ఉద్యోగం కావాలన్నాడు. వాడికి ఎక్కడో PhD చూపిస్తే ఈ చుట్టుపక్కల వాళ్ళు ఉండే ఈ హాస్టల్ చుట్టుపక్కలే, సార్ నాకు PhD వద్దు, నేను కంపెనీకి పోవాలంటాడు. ఇంకో కంపెనీకి పంపిస్తాను. వాడు మనం చెప్పకుండా ఇంకో కంపెనీకి వెళ్ళిపోతారు. ఇంకా రకరకాల వేషాలు ఉన్నాయి. ఎన్విరాన్మెంటల్ సర్టిఫికేట్ ఎట్లా వాడుకుంటున్నారో కూడా అర్థమైంది. ఇక్కడ నేను చెప్పకూడదు ఈ ప్లాట్ఫామ్. సో యూ హ్యావ్ ఎ లాట్ ఆఫ్ ఛాన్సెస్ యాజ్ ఎ ఎన్విరాన్మెంటల్ ఆఫీసర్ కానీ ఎన్విరాన్మెంటల్ కన్సర్న్స్ అనేది చాలా పెరుగుతుంది. ప్లీజ్ డూ జాయిన్ MSc Environmental Science. This is my request and appeal. Whatever you say that, and enjoy the research. Thank you so much, Savitri madam and other Ravi sir and a series of Ravi sars. I don't know, a series of Ravi sars and all the faculty, students. Please enjoy yourself in science. Please motivate yourself and use the opportunity wherever it is. Think of this. ఎందుకు మనం ఇది వినాలి, మనకి డబ్బులు వచ్చేదే కష్టంగా ఉంటుంది. Try to use this opportunity. And again, once again, thanks to all the degree college from the principal to Savitri madam. Thank you so much. And నేను కాదు, నన్ను ఏ క్వశ్చన్ అడగలేదంటే నేనేం సరిగా చెప్పలేదని అర్థం కదా.
Let's start to sir. If you have any queries or discussions, it's open for discussion.
మేడం ఛాలెంజ్ చేస్తూ చెప్పినట్టు చెప్పింది. కనీసం 2-3 క్వశ్చన్ అన్నారు. మీ అంతా చూస్తామన్నట్టు చెప్పింది. ఇప్పుడు దాన్ని ఒక్కరన్నా అడగండి. మేడం నేను పంపిస్తాను, ఎందుకంటే నేను అది ఎక్కువ చెప్పలేదు. ఇప్పుడు జర్నల్స్ అందరూ కూడా ఈవెన్ ఏదైనా రికార్డు ఉంటే పెట్టమంటున్నాను. యాజ్ వన్ ఆఫ్ మై క్లాస్మేట్ రాసేది కానీ పీలర్లో చాలా బిగ్ ఇష్యూ. అన్ని విత్తనాలు ఇప్పుడు కొత్తగా చేస్తున్న విత్తనాలు ఎప్పటి నుంచో ఒకసారి రాస్తున్నారంటే నేనే కొత్తగా సైన్స్ వచ్చి చెప్పాల్సిన అవసరం. విత్తనాలు ప్రొడ్యూస్ చేస్తాను. ఇట్స్ నాట్ ఎ బిగ్ స్టోరీ. స్టోరీ is hydrogen as well as the conversion of animal waste.
Creating electrochemical gradient in the membrane and helps for synthesis of ATP. Is it right or wrong?
100%. But not all the protons. Not all the You said is correct. It will be, it's a gradient in the protons and the membrane transporter. It will be useful. Not only this one, sodium, whatever, potassium, and all the gradient transporters. But one thing is not all the protons, H plus protons. Very few are working on that. Remaining, what will happen? If you go and crush it and go with the fermentation, hydrogen and sulfur is this and all one of the fermented product. That's what people are missing. H plus has its own role in plant cell, either including animal cells. what will have the remaining hydrogen? That's the story. Your observation is correct, but at the same time, we are thinking of bulk amount of protons. Water splitting is a continuous process in plant. One person asked me nicely, sir, you are telling this photosynthesis. When will plant will or algae will start the photosynthesis? I don't know. Even I don't know. Whenever the threshold levels, if too much, sir, he asked a very nice question, sir, you are telling like a photosynthesis respiration, both will be there. When will the photosynthesis start? When will create the stress? That's the way you need to ask that question. If algae has its own threshold limits, if sun is too much, it won't go for photosynthesis. Even CO2 is more in the, it won't go for, or even more contaminated water, it won't go for the photosynthesis. That is a flexibility. That depends on the algae also. Even some algae will go for that much of hydrogen, and other thing, depends on the algae and their habitat. Actually speaks habitat. Whether it is there in marine areas, whether it is there in the terrestrial areas, whether it is there in the minor. Yes, but yeah, it will not in the. I'm not sure that. I'm not sure.
One more idea is maybe in the sunlight, all the when photosynthesis process is occurring, all the H plus accumulates in the cell, and in the night and in the dark,
It will convert it. It will convert it. Even if you stop, if photosynthesis is there, then only oxygen will come.
Yes, sir.
If photosynthesis stops, oxygen will be not there, then automatically it converted into hydrogen.
Hydrogen is actually.
That protons converted it, naturally it was converted into hydrogen. You don't need to do any stress also.
Yes, sir.
That's what I said, even light, too much light or dark conditions. Walls are favorable conditions for hydrogen production. You don't do anything in night
By
time,
nature
automatically
itself.
hydrogen gas will form. That is the one of the, that's why we are getting hydrogen gas as a one of the fermented
Only
product.
thing is we need to capture the hydrogen.
But